Excellent. Right. So I think we'll start. So good afternoon, everybody, and thank you for joining us. We've had quite a long summer break in terms of our webinars, but I'm really delighted that today Adrian Fox from Ferra Science Limited is going to talk to us about the work on carrot virus. And I was inspired to ask him because he actually gave at least some of this talk at the International Congress of Entomology. And it sounds really interesting, both in the context of carrots and more widely. So I think most of you have done it already, but please could you turn off your camera and mute your microphone when not speaking. We're already recording the session and it'll be available later. I've applied for basis points and have had an acknowledgement, but they haven't come back to me yet. And just to say that the next webinar, which will be on the 4th of December, is about beans and is about creating a marketplace for new varieties of UK registered beans. And some of you will be aware that there's been a lot of research at Warwick and elsewhere to do with production of UK beans. So we'll take questions at the end of the presentation, but please feel free to type questions in the chat as we go along. Right, so if you're ready to go, Adrian, I'll hand over to you.
Thanks, Rosemary. I'll do the usual slide share and then just check everyone can see them.
I can see them.
Excellent. That's good enough for me.
Brilliant.
Okay, so good afternoon. For those of you that don't know me, my name's Adrian Fox. I lead the plant virus diagnostics team at Ferra Science in York. Our role is primarily as a biosecurity service, so we work with the Plant Health Inspectorate and we do the import-export plant health testing for England and Wales. But we also work in support to policy in terms of doing things like research projects to help inform whether a newfound pathogen is going to be a risk or not, but also working out how to better manage things that are either here or emerging and becoming a plant health risk. And quite often that means that we can't do field scale work on things like quarantine pathogens. So it's really useful in those respects to start playing around with things that are domestic problems, what we might call quality pests, to try and get a better handle on how we might deal with those. And then that can inform in turn how we would deal with a quarantine pest should it occur. So the talk I'm giving today, as Rosemary said, at least part of it was given at the International Congress on Entomology in Japan. And I spoke in a session there on the relationship between public policy and vector-borne diseases. And I thought, rather than going into how public policy may either cause a problem or help a problem, what I thought was interesting is to consider the interrelationship between public policy, policy change, and agronomic practice, and really how we as a science community can help to support growers and help to support policy in terms of making informed decisions, but also to help understand and mitigate against potential adverse effects from possible decisions. So you'll notice on the authors list there's myself and some of the people from my team here at Ferra, but also Andrew Dukes and Rosemary from Warwick, who did a significant part of the virus work that I'll be talking about. There we go. So thought we'd start on a carrot virus talk by firstly talking about potato virus, because that's always what you do when you haven't quite revised for your exams, isn't it? So what we're seeing is a general rise in what we call persistently transmitted viruses. And so these are the viruses that require an aphid to sit still for a long time because you have a circulative mechanism of transmission within the aphid. So the aphid has to have a proper feed into the vascular tissue. The virus is then taken up, passes through the midgut, and goes back into the salivary glands, where it's then ready for transmission again, and it has to have another proper feed for it to be passed on. And that's an interesting situation for us virologists, and probably Rosemary is an entomologist as well, because for most of my 30-year career, we've spent our time warning about the risk of non-persistently transmitted viruses. So those are the ones where you have a quick probe feed, the virus is picked up by the aphid, and then it moves quickly on somewhere else, has another probe feed, and can pass the virus on. And we'd spent a long time worrying about those viruses because they're the ones that are really difficult to control with chemical controls. Because if you've got something that is picked up by a simple probe feed and passed on by a simple probe feed, we've never had chemistry quick enough to knock down the aphid populations to get good control of those viruses. But what we've been seeing lately is a rise in these persistently transmitted viruses. And the opposite situation is very much the case with these, that if you can't get chemical control of a non-persistently transmitted virus, if you need a feed that takes minutes, you're talking it's for the virus to be recirculated and another several minutes for it to be passed on, then you actually have got a situation where you should be able to get good chemical control. And what we've got is a situation where what we're now seeing, if you look at the graph that I've been kindly allowed to use from SASA in Scotland, from 2017 onwards, if you look at this blue line, there's a pointer to work. There we go. So if you look at this blue line here, which is your incidence of leaf roll, you've got this kind of generally increasing picture. And I don't have the latest figures, but I'm led to believe it does carry on going up like that. And there's a lot of concern in the potato industry about this increase in leaf roll. And really, we haven't seen levels like this for 20 years, 30 years, and certainly before I came into this business. And one of the reasons was that we've had this effective chemical control. But we're seeing these same problems across a number of these persistently transmitted viruses. These are traditionally what we call the yellows viruses. So potato leaf roll virus in potato, barley yellow dwarf virus in cereals, turnip yellows in brassicas, and lately we've done some work to show it's also the predominant virus of concern in pea crops as well, and maybe causing up to 40% yield loss in those. And as I'll talk today about carrot red leaf virus again. And so there's potentially something happened in the past 10 or so years that's changed the landscape, that's allowed these viruses to become back to the fore and to start predominating again. Now, if you start seeing a change in 2017 and wonder about what may have happened, there's one obvious public policy change that occurred in 2017, 2018. But I think it's a gross oversimplification to just say it's banning neonicotinoids that have led to this increase, because we still have some neonicotinoids, and within seed potatoes you can still use a couple of treatments of neonics per season anyway. So it is a gross oversimplification to say it's just the banning of neonicotinoids. However, what we've done at the same time as reducing the use of the neonicotinoids, which were effective insecticides and did work well, is that we've also got a build-up of resistance within certain aphid species, and many of those aphid species, especially Myzus persicae and Cvararigopodi, as I'll talk about today, they are species which are transmitters of these persistently transmitted viruses. They are the colonizing aphids that we worry about. But we've also seen a change in our climate and our landscape usage across that time. And so I think all of these things working together are contributing to that increase in the amount of virus we're seeing. The other part of the story that we have to think about is actually, if we don't have effective chemical control and we don't have control over what's happening around our crop of concern, we also have to think about our agronomy. And traditionally we would have talked about cultural controls. So if you don't have a decent chemical control, you have to worry about your cultural controls. If you don't have effective cultural controls, then you really are in a problem, because you got no way to control the problem at all. And I think, as we'll show, that our carrot patho system in the UK is partly driven by the agronomy that we use. So unlike other areas of mainland Europe, we don't harvest our carrots in autumn and put them in boxes in store. What we do is we store them in the field under straw. It's more cost effective. We grow our carrots in a way that means that we can effectively pick fresh carrots right the way through the off season, but we're also picking them at a time in the year where we've got new carrot crops in the nearby vicinities, because you have a very limited number of regions where you can effectively grow commercial carrots at scale, because you need certain soil types. And so what you're then doing is you're pulling the straw off at a time where you have young crops next door. You may have a buildup of aphid populations under that straw, and if that's then heavily infected with virus, it can take straight off and affect crops next door. Traditionally we would have worried about the weeds that are part of the patho system as well. And we often talk about this spillover, spill back system, where in the autumn, if you lift a crop, so say potatoes, carrots, whatever, then your virulous aphids, your virus carrying aphids will move on to that, those hedgerow weeds. They will overwinter. If you know anything about the Apiaceae, so umbelliferous weeds, you have this kind of constant churn through the season of different weeds from that genus. And most of those will be susceptible to the same viruses as carrots. So you could be getting this system where you're going from weeds to carrots and back again. And where we first got involved with carrot work was back in 2012. One of the growers came to us with this problem that was causing this internal necrosis down the core of the carrot. And what we were seeing was that in quite a low percentage, but enough that an entire crop would get rejected, you were getting this internal browning. And the grower and agronomist involved were pretty sure this was a virus problem, because it always happened in crops where you'd had high amounts of aphids and high numbers of virus. And we managed to link it to this virus called carrot yellowleaf virus, where, again, not every plant that was infected led to this, but it looked as though there was a relationship between time of infection and the amount of time the carrot crop had been grown for after that point of infection that was then leading to this necrosis. And that led us onto an arc of carrot virus work, where we were then trying to understand what was transmitting this virus, and largely focused on carrot yellowleaf to start with. But we also discovered a whole bunch of other carrot viruses that at that point we didn't even know existed. And so that then led us onto an arc of research of trying to understand the epidemiology of these and using them almost as models for how do you deal with a novel virus, and you need to try and risk assess that virus within the system. So the first thing we did was to try and understand what the predominant carrot viruses were in the UK. And so we did a few field studies, and this was work of a former PhD student, Zarina Rosado, who was based with us at Ferrer. And she went to some fields in Yorkshire near to Ferrer, and some fields down in Norfolk. And you'll notice I've quite handily used a little house, because Yorkshire's where I live, so that seemed to be the appropriate icon, and a little star for Norfolk, just in case nobody knows where that is. And so what we found was that actually the predominant virus was what we call the carrot motley dwarf complex. And that's not a single virus. I'll talk a little bit more about the epidemiology of that in a moment, but it really was the predominant virus. But what we did see is the new virus that we were worried about, so carrot yellowleaf virus, was certainly in some fields, so locally, was as important as the carrot motley dwarf complex. And then we also had this other new virus called carrot tornado virus. And again, that seemed to be more of a problem in some regions than another. And we had this other virus as well, another novel virus from the clostrovirus genus. And so what we could then say is, well, actually, even though we're getting multiply infected plants, so this was testing 100 plants per field, so you've got at least two viruses per plant within this crop of hymen infected here. But interestingly, in the crop that was thought to be healthy, we also had more than 100% virus prevalence within that crop. So what we could say is that the viruses really were a major constraint on production, and the carrot motley dwarf complex was our major viruses of concern, but all these other viruses would also be contributing to virus problems that we were seeing. And so once we'd worked out what the major viruses were, we then did some work looking at trying to understand the epidemiology of these viruses. And the first part of that is looking at what aphids are transmitting them. And we'll come back to some more work that we've done on this. And initially, this was all laboratory-based studies looking at the relative efficiency of transmission. And so, as I said, the main virus of concern, carrot laboratory dwarf complex, is actually a complex of multiple viruses. So you've got the carrot red leaf virus, which is one of these persistently transmitted polariviruses. But what you also have is a second virus and a viral RNA that are encapsulated within carrot red leaf virus. So when a virus encapsulates, it's not a specific action. And so if there are any other small RNAs that are kicking around within those same cells as that virus encapsulates, they can effectively be wrapped up within that same protein coat of the virus and then transmitted on as a hitchhiker. Think of it as a Trojan horse, if you will. And so we showed that Mysis persicae was a transmitter within carrot, and that came out in an AHDB report. And about the same time, Nassim, working in the Salinas Valley in California, also showed that Mysis persicae was a transmitter of carrot red leaf in parsley crops. In the novel viruses, we showed that this Mysis persicae was transmitting the carrot yellow leaf virus, because previously we'd only looked at the Cavariellas, and that was work done back in the 1970s up at the Crop Research Institute in Dundee. And then the Torrado virus, we showed that both Mysis persicae and Cavariella erygopodi may be transmitters, and we also showed that it was semi-persistent transmission. So what we've now got is multiple different virus transmission mechanisms going on. Some of which take a lot longer than others, some of which require helper viruses, some of which are encapsulated with viruses. And so previously we thought we had maybe one or two viruses of concern. And what we then realized was we had several viruses and the system was a whole lot more complex than we'd actually given credit for. So the next question was actually to look at those alternate hosts. And so, as I said, we traditionally think about viruses in a spillover, spill back type relationship. And we were really fortunate that the late Des Barbara, who had been based at Warwick, had been doing a project looking at carrot red leaf virus, among others. And he'd collected over 3 years over 1,500 plant samples from across the UK and had put together this kind of phylogeny of them. And what he'd shown was that you kind of got groupings of the different virus isolates depending on coat protein sequence. And what you found was that you had some that were largely carrot, wild carrot, some that appeared to be restricted only to cow parsley, and some which had many wild hosts but very few carrot. So that then suggests that you've got a slightly different epidemiology for some of that carrot red leaf population from the other carrot red leaf population, and they may in fact be circulating independently of each other. So if you want to think about it, you've got a carrot red leaf population and a weed carrot red leaf population. But we dug back into these other 1,500 isolates, and what we managed to do was test them for the novel viruses, so carrot yellow leaf virus, carrot clostro and carrot Torrado virus, that at the time that Des was doing his work, we didn't even know these viruses existed. And what we showed that cow parsley was probably a major environmental host. And I'm not saying reservoir, I am saying host intentionally, because we don't have that direct link between the host and the virus. And hogweed was possibly playing a minor role as well. But interestingly, for the clostrovirus, there was quite a bit more of it in the weed hosts than the other viruses of concern, which would suggest that that may actually be, if you want to consider it, a weed virus rather than a carrot virus, and then it's inadvertently been found in carrots. So we know that some aphids have the potential to transmit these viruses, and we know that they are in some hosts. But what we don't know is which of those main vectors we should be worried about. So we know that for carrot red leaf virus and for carrot yellow leaf virus, both Mysis persicae and Cavariella erygopodi can be potential vectors. But what we don't know is what's actually driving that transmission. And so we got an AHDB project. So Rosemary, myself, Andrew Dukes, and the rest of the guys here at the ferrovirology team. And what we wanted to do was look at the timing of transmission of carrot viruses so that we could try and improve the management strategies. So one question is it multiple aphids that are transmitting those viruses? Are they coming in certain waves? Is that season-long transmission? Does it happen for a short period of time and then stop? And if we can identify which aphids are the main vectors, and then what the peak times for transmission are, then can we give advice on how to better target treatment regimes? So if you've got a restricted amount of chemical you can now use, then actually by better targeting that, you can probably get a better lifespan of the chemical without resistance building up, but also it becomes more efficient to then think, well, actually, after such a point, I don't need to keep spraying anymore. And so initially, we had replicated trials at Warwick and in commercial crops at Ferrer, and this was basically an uncover and recover trial. So plots out in the field with aphid-proof netting dug in, and on a weekly basis, a plot was uncovered, a yellow water trap was stuck in the middle of it. We caught whichever aphids were moving into the crop at that point on the yellow water traps. There were also aphid counts in the field. After 1 week of exposure, that plot was covered back up, and so then any virus that was in there was effectively limited for further spread, and then you would move on to another plot. And after 4 weeks, they were uncovered, and 100 plants per pot were tested, and they were tested for red leaf and yellow leaf by qPCR up at Ferrer. So we started this in 2019, and the first season went really nicely. You'll notice that I've only got the University of Warwick results here, largely because the Ferrer ones in a commercial crop, we got very little virus transmission, so there's nothing we can really correlate between aphid movements and the virus presence from that. But what we did see was that you got this kind of initial early season transmission, which then kind of died off a bit. We think you're getting some effect here as you're pulling, as you're uncovering crops, as you're covering plots, you may get slightly higher incidence, but that pattern of transmission is still being driven largely by Cavarilia aegopodi. So you get this little rise in aegopodi numbers here, and you get the corresponding rise in aphid in virus transmission. 2019, we felt quite confident that we could say that Cavarilia aegopodi was driving that transmission, certainly of carrot red leaf. But as you'll see also for total virus, there was very little carrot yellow leaf moving about, so it's difficult to actually draw any firm conclusions on that. And then 2020 happened, and everyone worried about viruses that weren't carrot viruses. Oh God, Boris Johnson, I thought he'd gone into the past. Anyway, so we postponed field work for 2020. The only good thing that came out of 2020 as a virologist was that I no longer have to explain to anybody what an LFD is or what a PCR test is. But I think the least we say about COVID, the better at this point. So we resurrected our trials in 2021, and what we saw was certainly in the early season, we got that same rise in Cavarilia aegopodi numbers, and the rise in virus transmission kind of mirrored it. However, if we look at the late season, we got a second peak, which did correlate with our total aphid numbers, but didn't have any correlation with Cavarilia aegopodi. However, what we did find was that this second peak looked to be associated with Cavarilia pastinaci, the parsnip aphid. So again, what that suggests to us is that certainly that early season transmission is largely driven by the movement of Cavarilia aegopodi, but there may be other aphids at play later in the season, and in some seasons you may get these secondary peaks. One thing it is key to notice is the difference in scale between the 2 years. So 2019, we got less virus transmission and much fewer aphids. There were about 5 times more aphids in 2021 in our traps than there were in 2019. But again, we're looking at correlations within year rather than directly comparing between years. So in conclusion from those field trials, as I said, minimal virus transmission at the York site, and it's difficult to draw any conclusions over what was going on. But we got greater transmission in aphid numbers in Wellesbourne in 2021 than in 2019. Similarly to 2019, the virus transmission tracked aphid numbers really well for the first half of the season, but then that kind of fell apart a bit late season. But as I showed on the previous graph, you got this late season peak of pastinaci, which did correspond really well to that late season peak of a virus. And so, as I said, what we think is that you've got two potential virus, two potential vectors at play within this system, and then you need to start thinking about within the epidemiology whether you're looking at early season problems or late season problems. And so the final year we switched to a spray trial based at Wellesbourne, and it was looking at the effectiveness of different treatment regimes. And I think the first thing we could say is that certainly in terms of yield, any treatment is better than no treatment, which is good news for everyone. But we did look at both virus levels and disease levels, and disease was measured by yield. And what you could see was that in the treatments where you had early season focused control, so some of them we only had treatment at the very start of the season, you got really good virus control early on. And then you the control broke down later in the season. And so those early season focused treatments gave better early control of virus, but that virus then increased later on. And the general picture was that early season control actually gave better disease management. So not only did that early season control give you good virus control early season, but it also led to a correspondingly higher yield at the end of the season. So later on, you may get virus buildup, but you've allowed your crops to get past a point where they are where they're susceptible to virus impact rather than virus infection. And so one thing you have to think about within that yield modeling is, are you aiming to protect your yield this year, or are you aiming to minimize inoculum and then spread for future years? And that leads to a really interesting, almost philosophical question about, do you go for bang for your buck in this year, or can you actually manage to try and control future virus levels on an ongoing basis? So at this point, I'm going to take a slight left turn and talk about another project, but I will bring it back to the virus work in a moment. And so halfway through the virus management work, we were funded by BBSRC as part of their bacterial plant disease program to look at Candidatus Librobacter solanacearum. And so that's an insect vectored phytobacterium, and it also affects potato, tomato, but it does affect carrot and celery. And actually within Europe, the majority of the problems that we see, we think, are within the Apiaceae rather than the potato issues that have been seen elsewhere. There are multiple haplotypes. There's one known to be present in the UK, but we have no idea how widespread this pathogen is. Certainly within carrot crops within the UK, we doubt that we've got the potato haplotypes because we do annual surveys of potato crops and we never see it. But we know there are multiple psyllid vector species that are present within the UK, and there are also species which may be vectors. And so we put a project together with ourselves, Sasa, Newcastle, Strathclyde, John Innes, and Rothamsted to look at the presence of Candidatus Librobacter solanacearum, the potential vectors, but also understand what the potential impact of this, and to try and understand why in the UK we don't see the major issues with this that are seen elsewhere in Europe. And so we were essentially asking a couple of key research questions. So why isn't the pathogen observed at the same instance in the UK as it is elsewhere? And what changes might lead these benign low-level infections to become damaging epidemics? So again, the pathogen we're thinking about here is also known as the zebra chip pathogen, and first reported in New Zealand, then in the USA. And in both New Zealand and the USA, it's predominantly an issue in solanaceous crops, tomatoes, potatoes. But in Europe and Northern Europe, we tend to think of it as a problem more on the apiaceous crops. And so what we wanted to do was to see whether we have the pathogen here, whether it's present within wild hosts as well as within apiaceous crops. And then we could try and understand the epidemiology of it and try and understand whether those changes in land management or changes in public policy might actually drive an increase in the amount of Librobacter that we see. So to do this, we followed an ecology-based sampling strategy. So we were taking paired transects around fields, and we were also doing transects across the middle of the fields. We were stopping every 12 meters and taking quadrat sample. So within the crop, that was 5 carrots per quadrat. Within the margins, we separated into target species. So that was mostly the apiaceous species I talked about earlier. So cow parsley, hogweed, Alexander's, rough chervil, things like that. But we also looked at things like nettles, where we know there are haplotypes, and we looked at roseberry willow herb because a previous study in Scotland had found that the psyllid, which lives on roseberry willow herb, was found to have a Liberobacter within it, and so we wanted to look at those as well. And so, to cut to the chase, rather than taking you through all of the research, what that actually showed is that we don't really have any issues at all with Liberobacters in carrots in the UK. Within the vector species that were caught within carrot crops, there was a low level of the nettle psyllid, which did have LSO, but we think that was just transient through the crops. The two that are likely to feed on carrot and apiaceae, we found no Liberobacter present within the vectors. And if we look at the hosts as well, we did find, again, the nettle psyllid had quite a high incidence of Liberobacter in it, and so did stinging nettles, to the point that actually for the second and third year of the project, we didn't actually sample any stinging nettles because we had so many positive individuals from the first year. We didn't find any within our apiaceous weeds, but we did find a couple of percent within the Anthryscus psyllid that was taken from those hedgerows. And again, the roseberry willow herb, we found high-ish incidence, but that was against really low numbers of psyllids that were caught. And we think what this is that we have a different land use here within the UK to places like Scandinavia, where Liberobacter is more of a problem. And the psyllids tend to overwinter within sorry, mental block. Tend to overwinter within pine forests. And we don't really have that many pine forests in association with carrot crops nearby. And we think that's one of the reasons why we don't see very much of the pathogen, because we just don't have the vector populations to support it. But one thing we did do was we took all of the samples that we collected from 4 of those caliber fields, and we resequenced them for the presence of the different viruses. And what we found, this is just a very brief excerpt of the known viruses that we found. There's a whole bunch of other novel viruses as well that we discovered. And what that allows you to do, because we've taken those samples using an ecology-based sampling strategy, we can then use network ecology to try and work out what the interactions between those different hosts and viruses are, and to see whether that can then inform what our possible vector pathways are. So the first thing we can say is that the results that we found for carrot red leaf virus actually do support the work that Des Barbara did back in 2010, 2012. And we found those very same patterns within the carrot red leaf, where you have basically a carrot circulating population and weed circulating populations. However, by doing whole genome sequencing, what we also found was it gets a whole lot more complicated, because they look to be about 4 different genotypes of virus, and they don't clearly link up with those different groups. So there's now recognized carrot red leaf, wild carrot red leaf, Pterolis crimson leaf, which are all very closely related to carrot red leaf virus. And they again have, they don't quite cleanly match that same coat protein sequence analysis. And further recent work from ourselves, from France and from Spain, has shown that the carrot virome is way more complex than we actually appreciated. And there are now dozens of viruses at play within that virome that we didn't know existed. And there seems to be a difference in distribution between different apiaceous hosts. And for a lot of them, you have to go to the sequence level. So if you have a simple, is this virus present, you will find it in multiple hosts. But if you go to the sequence level, as we've done with the carrot red leaf, you'll notice that there's actually a separation and a host specificity within there. And the other thing we can do, and we're in the process of writing this up at the moment, is we can use that virus ecology network analysis to work out what our pathosystems look like. And so the viruses I talked about, which were novel back in our 2014 study, the carrot clostrovirus and these carrot caudoviruses, we've got no prior biological characterization for these. We don't know what their their individual pathosystems look like. But what we can say from here is that actually, if you look at the carrot clostrovirus, there seems to be a very tight link between carrot and cow parsley, and the same is true of the cordoviruses. And so one question there is, have you got the same aphids driving the transmission of those that we see for things like carrot red leaf virus? And so where we stand at the moment is we still have a lot more to learn and a lot more to work out. But I think if we can use every tool we have available to try and better understand the pathosystems we're dealing with, it puts us in a much better position to be able to advise policy and advise growers on what changes need to be made and how better to manage the problems that we see before us. So a few acknowledgements, people that did the work: Innes, Mary, Amy here, Andrew Dukes and Rosemary at Warwick, Fiona Hyatt for the loan of the potato data, the people that helped support us through the carrot work, so Howard, Roger, Ian and the British Carrot Growers Association, and for the HDB and UKRI for funding the work. Thank you for listening.
Brilliant. Many thanks, Adrian. Gosh, that's packed with information.
It was quite a lot to fill in.
Loads to go at. Right, has anybody got any questions for Adrian? So please put your hand up and you should come to the top of my list, and then or turn your microphone on and shout. No? Okay, so I'm going to ask you one anyway. So I think we know now that willow carrot aphid Cavariella overwinters in two ways, which way they use might depend on geographical location as well. So they can overwinter as eggs on the woody host, and they can also overwinter, I guess, if the weather's kind, as mobile aphids. How do you think that, knowing what you know now, how do you think those two modes of overwintering might influence virus transmission?
So I think certainly early season transmission, I suspect, is coming from what you might call those aphids that have been on vegetative sources rather than tree sources. And also I suspect that that's where you're getting virus carried from. So if the aphids that are coming from willow as a result of sexual reproduction in the previous season are going to have to land on something to pick up the virus before they move into a crop, whereas the ones that are coming from overwintered crops are potentially carrying virus already. And I think they're more likely to start building populations and get that jump on the season. So my suspicion, given what we're seeing from that work that was started by Des, and we've complemented with what we've done, I think there is a suspicion that it is those aphids overwintering on vegetative sources, whether it be hedgerows or under straw, that are initially driving that issue.
Okay, I just wonder how that sort of ties in with suction trap captures, and whether the suction traps catch both winged aphids from both overwintering types. I have no idea, but
no. It's an interesting study that somebody could fund if we can use the F word at this stage of the day.
Brilliant. Okay, so Roger's got a question. Roger, do you want to turn your microphone on?
Thanks very much, Rosemary. Hi, Adrian. The question is about 2024, when you've seen the consistent massive increase in virus over since 2017, and then in 2024, unexpectedly, aphid numbers. This is Cavariella agropodi numbers and virus and virus within the carrot crop have collapsed. Is it a complete one-off, and why has it happened? What are your thoughts on that?
I mean, the interesting thing is that we haven't necessarily seen a similar thing with other crops. So from a potato perspective, we've continued to see, maybe not as high levels of aphids, but certainly that virus levels have continued to drive through this year. We didn't have an overly harsh winter. That's normally the first place that we'd go and grasp for a solution. If you've got kind of a late snap of cold, that can knock the aphid numbers back. So I think it's possibly a one-off, and I would be nervous about whether we'd see that again. But as to the why, I think there's a lot of questions there, Roger, and I don't actually have any answers.
My own gut feel is the incredibly wet spring was the notable thing about 2024, and I wonder if it's just washed them away. But
yeah, and again, as there's also a question. I mean, judging by, I know that you're not a million miles away from me here, but judging by the fact that around this area we're built largely on sand, and most of the golf courses were shut since about October. That would suggest that there was a lot of water falling right the way through that winter. And it could well be one of the reasons that, as you say, if they're too busy swimming, they're not going to be getting a chance to bulk up numbers.
Yeah. Thank you.
Yeah, I mean, I think a sort of a number of species that we value, for example, butterflies, have also been really hit this year. And the explanation from butterfly conservation is, again, the wettish sort of winter come spring, and then some conditions. Well, the summer wasn't very good either, which have really sort of whammied some of the species. So yeah, any more questions? Adrian? No? Okay. Well, there's a long message for you and for me from Priska. Yeah. Oh, you've replied to her. That's all right. Or them. That's all right then. But yeah. Right. Okay.
There's one from Charlotte.
Oh, Charlotte. Right. Hang on.
Just asking about seed-based propagation of Candidatus liberobacter. I mean, I think the question there is, okay, so we've looked at carrot because there was a report of seed transmission of liberobacter in carrot, and does that happen in field margins within the weeds? My gut feeling is it's still not a major concern within the epidemiology of that pathogen. Certainly the work that was done that initially suggested seed transmission has been difficult to replicate, and several people have tried it. And again, that's based on carrot, but I think you've got the same problem within just the mechanism of seed transmission is going to be quite hit and miss anyway. Even with things like viruses that will go properly into the seed, it's not a clockwork 100% seed infection relationship. So I think even if it does happen, I think it's going to be at such a low level, it doesn't give a major concern by comparison to that moving from, say, apiaceous weed to apiaceous weed through the season. And as I say, that overlapping of different species that will allow that green bridge to go right the way through the season, I think is, to my mind, one of the bigger concern for that.
Excellent, thank you. Right, if there aren't any more questions, then just to say very many thanks again, Adrian. Really interesting talk, and I'm sure I'll come back to you with some more questions afterwards, because it's all quite complicated, isn't it, really?
Yes.
And thank you everybody for joining us, and hope to see some of you at the next webinar in December. So thank you very much, and have a good evening.
Thanks, Rosemary.